Hi everyone, this lecture is about bioproduction of polylactic acid Suture, and we are students from Washington University in St. Louis. Our team members include Zhenqing Wang, Lei Rao, and Yihang Yuan myself. Here is the content. There are mainly 4 parts. Firstly, we will introduce the concept of suture, lactic acid, and polylactic acid. And then we will move on to the synthetic methods of polylactic acid. We also include the economic analysis of a case study. The final part will be the conclusion. So firstly, let's look at what suture is. By definition, suture is a kind of biomaterial device, either natural or synthetic, used to ligate biovessel and approximate tissues together. Here from the right-hand side, the figure shows how suture works in the surgery. As an ideal suture, it should be easy to handle during surgery. It should elicit minimum tissue reaction, no allergic or carcinogenic reaction. It should not support bacterial growth. More importantly, it should possess high tensile strength and easy to sterilize. It would be even better if it can be absorbed after severing dysfunction, so that no second surgery is needed. And then we can look at the classification of suture. Generally, suture can be divided into 2 groups based on its source. One is natural and the other is synthetic. It can be further classified based on whether it can be absorbed. Here, in this lecture, we will mainly focus on polylactic acid as an absorbable and a synthetic suture material. Then we will introduce the polymerization process, properties, and biomedical application of polylactic acid. Here is a figure showing the polymerization process of polylactic acid from lactic acid monomer. Lactic acid is a naturally organic acid that can be produced by fermentation of sugars obtained from renewable resources, such as corn or sugarcane. By polymerization process, it can produce polylactic acid, also called PLA. The properties of PLA are summarized in this table. We can see the density of PLA is 1.21 to 1.25 gram per cubic centimeter. The tensile strength of PLA is around 21 to 60 times 10 to the 6 Pascal, which indicates its extensive mechanical strength. So here, we can see that PLA is a thermoplastic and biodegradable material with high biocompatibility. It can be easily degraded by enzyme and hydrolysis. It also has extensive mechanical strength and good process ability. Because of these properties, PLA can be widely used in biomedical applications such as drug delivery system, implants for biofixation, and surgical applications such as sutures. Then she will continue to talk more about the synthetic method of polylactic acid.
So as we look at the synthetic methods of PLA, they can be divided into 2 categories. The first one is chemical categories, and the next one is biological methods. So for the chemical methods, we have 3 different methods. And the first one is directly use the biocondensation, and we can generate low molecular weight pre-polymer. And if we continue to get catalysis, we can get PLA at the end. The second one is about isotropic dehydration, and this is kind of a direct method of synthetic and for high molecular weight of PLA. Basically, this is the removal of water. And third one is ring opening polymerization, and this is the most common route for achieve high molecular weight PLA. The process consists of 3 steps: polycondensation, depolymerization, and ring opening polymerization. By controlling resistance time and temperatures in combination of catalysis and concentration, it is possible to control the ratio and sequence of the lactic acid units in the final polymer. So for the biological methods, if we use glycolysis, the fermentative pathway, we can generate lactate. And for PLA synthetic methods, we can use recombined E. coli strains and use and combine with sulfurous like PAC-CP gene. So if we look at specifically for lactic production, we can use fermentative pathway and glycolysis. For fermentative pathway, we can generate 6-PG and G3P and eventually get lactate. For glycolysis, we can generate F6P and G3P and generates lactate in the end. So if we look at this metabolic flux analysis, we can use silos from lignin cellulose and use different bacteria and yeast to generate G3P and pyruvate, just as glucose did. And if we use different dehydrogen anions, we can use LDH and LDHA, like L-lactate and D-lactate. However, what we want is actually the D-lactate because that's much stronger polymer and higher heat resistance. So if we look at PLA production, if we look at the glycolysis pathway, we can generate glucose, and we can generate lactate from glucose using LDHA. And if we use E. coli, which is the GLX7 strain, and combine with the transfers, we can generate PLA at the end. So the next part is actually the economic analysis.
As mentioned before, PLA is very important in medical applications, and its monomer lactic acid is an important chemical substance with a large number of commercial uses also. They can be fermented and produced from biological raw materials. A previous research analyzed the technical and economic feasibility of a biorefinery to produce 100,000 metric tons of lactic acid from corn grains per year. And this study estimated the resources, including equipment, raw materials, energy, labor requirements, and cost to produce lactic acid from bacteria, fungi, and yeast-based fermentation pathways. For feedstock, yeast-based fermentation pathway required the lowest feedstock quantity due to a higher glucose to lactic acid conversion rate, but the fermentation step required similar amounts of nitrogen sources for all three pathways. The nutrient requirement was slightly higher for the fermentation pathways using bacteria and yeast than fungi. We can see from the left side graph the significant factors that affect the lactic acid production are sugar to lactic acid conversion rates, grain price, plant size, annual operation hours, and the potential use of gypsum. A major fraction of the raw materials cost was for producing the corn grain. Corn grain cost contributed most to the lactic acid production cost associated with feedstock preparation. In addition, for bacteria and fungi-based pathways, cost for lime and sulfuric acid used during the fermentation and lactic acid recovery process contributed to the raw material cost too. Raw materials cost for the yeast-based pathway was lower than the bacteria and fungi-based pathway, since it didn't require chemicals such as lime to neutralize the lactic acid during the fermentation, and sulfuric acid and water to recover the lactic acid from the neutralized solution. Also, the cost of product separation and recovery is the lowest for the yeast-based pathway because it reduced the utility required for distillation and eliminates the need for gypsum formation and removal processes, which also reduces chemical requirements and cost. So in this condition, the production cost of lactic acid from corn grain is about 1,181 per ton for bacteria-based pathway, 1,251 per ton for fungi-based pathway, and 844 per ton for yeast-based pathway. Besides, for the capital cost investment, all the pathways require the same size, type, and number of equipment for feedstock preparation, pretreatment, and fermentation steps. The fungi-based fermentation pathway required an additional air compressor and filtration unit to aerate the fermentation process. The yeast-based fermentation pathway didn't require lactic acid neutralization, and so the reactor for recovering lactic acid from the calcium lactate and the filter for gypsum removal was not necessary. The yeast-based pathway didn't require the lactic acid neutralization, recovery process, and the investment associated with the purchase and installation of equipment needed for this process. So the yeast-based pathway will have a lower total capital investment. The capital investment for the fungi-based pathway was the highest because this pathway requires a continuous supply of air for effective fermentation using fungi, like just as I mentioned before. It means an additional air compressor and filtration units will be needed. It will increase the capital investment cost, and the yeast-based pathway requires lower working capital due to the reduced chemicals, utilities, and labor requirements because of the for the lactate acid neutralization and the recover steps. So in total, the total capital investment for a 100,000 ton per year lactate production facility is about 130 million for bacteria-based pathway, 147 million for fungi-based pathway, and 130 million for yeast-based pathway. Now let's look at the polylactic acid global market. The US is the largest exporter of PLA in 2016. It exported PLA worth US dollar $116.47 million that year. We can see from the left side figure that the price of PLA is the lowest in the US in 2017, which ranges from $1,860 to $2,010. By the end of 2020, the global PLA market is more than US dollar 5 billion. And we can let's look at the right side figure. Even though the production of PLA doesn't account much in the production of bio-based polymers, there is still an increasing trend from the year of 2018 to 2023.
So as a conclusion, PLA Suture is a good, absorbable, and environmental friendly biomaterial for many medical applications. PLA synthetic methods can be divided into two categories, which is chemical and biological pathways. And advantages for this Suture is actually good biocompatibility, biodegradability by enzyme and hydrolysis, and a very strong, extensive mechanical strengths. However, it has their disadvantages, which is lower glass transition temperature and very rigorous control of synthetic conditions like temperature, pressure, and pH. For our economic analysis, PLA production capabilities grow annually in the global world, especially for yeast-based synthetic pathway. So that's all we have for our presentation. If you have any questions, comment down below. And don't forget to thumbs up and subscribe to the channel. Thank you very much.